Okay, Biology 107 fans, here's the Flip lecture for today. We're going to talk about proteins, what their structure is and what their function is. And what you should know so far is that we've talked about some hypotheses and theories. In particular, we've talked about the cell theory and that all living organisms are made of cells. And then we also talked about the theory of natural selection, Darwin and Wallace. They made observations, and based on those observations, the survival of the fittest was the process of natural selection, which explained the different beaks that they envisioned or saw on the different birds. We also talked about how RNA can be compared in organisms to develop a phylogenetic tree. And then we started talking about the molecules, the actual atoms that make up all of our chemical compounds. And we began to talk about energy and the conservation of energy, what kind of bonds get used. And now we're going to specifically address, in this lecture, we're going to specifically address proteins in a more detailed way. In later lectures, we'll talk about nucleic acids, carbohydrates, and lipids and what they are made into. So we talked about building blocks of life. So you had the 4 atoms. So you had hydrogen, oxygen, nitrogen, and carbon. And these 4 atoms get made into compounds. And the one that we're going to talk about today are amino acids. What's the amino acid? Well, that was the compound that was constructed of an amino terminus. And what an amino terminus means is that it has a amine group in it, a nitrogen, and it actually has 3 hydrogens on it. And a nitrogen, how many bonds should a nitrogen make? Well, a nitrogen total usually makes 3 bonds, and in an amino acid it actually is going to make 4 bonds. So it makes 3 to hydrogens and 1 to a carbon, and that results in this amino group. So it's called an amino acid because this is an amino group with a positive charge. Now, the central carbon in this chain is linked to the nitrogen by a covalent bond, which means they're sharing the electrons. You know that nitrogen is negative, so it has these electrons are closer to the nitrogen than the carbon. And then the carbon in the middle is linked to another carbon, and that carbon is linked to 2 oxygens, and this particular structure is called a carboxyl group. That's really important. You're going to see carboxyl groups all over the place. The double bond to an oxygen makes this oxygen on the top complete, whereas a single bond to the oxygen on the bottom results in the bottom oxygen actually having a negative charge associated with it. So this is actually the acid part of an amino acid. It has an amino group and it has an acid on the other end. So those 2 are always on an amino acid, and then we know how many bonds should carbon have. Carbon should have 4 bonds, excuse me. So 1 bond is always going to go to a hydrogen, and then the other bond goes to what we call an R group. And the R group can change based on which amino acid we're talking about. There are 20 amino acids. There's actually 22 or 23. Some of those are modified amino acids, but there are really 20 primary amino acids. And it's these amino acids that get put together to generate proteins. And the structure that links one amino acid to another amino acid is called a peptide bond. right? It's going to be a covalent bond, so it's sharing electrons, and it stores energy. This particular slide shows two ways of writing amino acids. On the top, you see what's called the non-ionized form. And in its non-ionized form, the nitrogen, which is always drawn on the left, only has 2 hydrogens associated with it and no positive charge. And the carboxyl group on the right actually has a hydrogen associated with it, and the oxygen now has 2 bonds, so it is not negatively charged. But really important is the fact that at neutral pH, which you are at neutral pH, most of you is at neutral pH except for your stomach and some places on your skin where you have sebaceous secretions, these amino acids always are charged. So you always have this positive charge on the amino group, and you always have this negative charge on the carboxyl group. So I would ask for you to draw for me amino acids using this particular ionized form, because that's what really happens in your body. There are lots of different amino acids. I do not ask Biology 107 students to be able to draw any of these. What I do ask is that you understand the different classes of amino acids, which are shown here. On the top, if you look at the green, which are the side groups, what I hope you'll see is that there are a lot of hydrocarbon chains. So up here we have mostly hydrocarbons. And hydrocarbons, especially saturated hydrocarbons, all of these, right, are hydrophobic. They want to stay as far away from water as possible. They don't bond with water. Water is polar. You know that oxygen has a partially negative charge, and hydrogens on that oxygen of water have partially positive charges. And these R groups don't want anything whatsoever to do with water, so these are often buried inside of a protein complex. Okay. The next group down, the purple ones, are polar. And I hope by now you know polar means that it's going to either have an oxygen, a nitrogen, or a sulfur on it. Alright, and so you see there's an oxygen, oxygen, sulfur, oxygen, nitrogen, nitrogen. And what that means is that there's partially positive and partially negative charges associated with all of these amino acids. And so those guys really do want to be in water, because their partially positives will interact with the partially negative charge on oxygen and water, and the partially, what did I just say, partially negative charges on these amino acids will want to interact with the partially positive charges on water, etc. And then the last 2 groups down here are actually charged, and one set is acidic, acidic, acidic. Is that the right way to spell acidic? It might just be acidic. AC. Spelling is hard. So acidic. And these guys over here are basic. What does that mean? These guys have negative charges, these guys have positive charges. Acidic means that they're donators of protons. So they donated protons, that's why they're negative charge on the oxygens here. And these guys have sucked up protons, so you see extra charges associated with this ring structure here, and this nitrogen and this nitrogen. And so if they're charged, they definitely want to interact with polar things, especially water. I don't ask you to know the structures, but in Biology 380, you do have to know the structures. You have to be able to draw them. You need to be able to draw the NCC with the carboxyl group and the amino group. So you have a negative charge and a positive charge. You have a hydrogen on the central carbon, and it's linked to the R group. That's what you need to be able to draw. and you should be able to recognize the R group's names. So the simplest R group is called glycine. It has a GLY or G as its 3-letter and 1-letter symbols, and the R group is actually just a hydrogen. There's another really simple R group for alanine, which ALA, ALA is the 3-letter symbol, A is the 1-letter symbol, and it is just a CH3 group, etc., etc. So I'd like you to read through the names of the amino acids so that you can recognize, if you see it again, that it's an amino acid. Okay, so the hydrophobic amino acids consist of these hydrocarbon chains. This is glycine, alanine, blah, 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 blah. Okay, tryptophan. I have to do these in their particular order. That's how I remember them. This is phenylalanine. This is methionine. This is proline. Then there's leucine, isoleucine, and leucine, isoleucine. I think this is leucine. I think this is isoleucine. And what is this one? Let's go back 2 slides and find out. Aha! It's valine, and how are you going to remember that? It looks like an upside down V. Oh, that's so simple. Why did I not just know that? Alright, so you should recognize these letters. You should recognize the names. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine. Notice it's not a P, but it's actually spelled phenyl, because there's a phenol group. Okay, tryptophan and proline. The polar charged groups that are acidic and basic are aspartic acid, ASP. A was already taken, so the 1-letter symbol is D, and glutamic acid, GLU, and the G was taken for glycine, so now you have D and E. And then the basic amino acids are lysine, K, because the L was already used for leucine. This is arginine, R, but it's really ARG, arginine. And this is histidine. Alright, so they're charged, there's the charge on this one, here's the charge on this one, here's the charge, here's the charge, here's the charge. These are the acidic and basic amino acids. Okay, well, when we start linking together amino acids, think about what is happening to entropy. Entropy, is this concept of disorder. So is disorder increasing or decreasing when you start linking amino acids together? So here you have an amino acid, and here you have an amino acid, and here you have an amino acid. And when you start linking them together, you go from disorder to more order. So if these start getting linked together now, you have a chain. Each individual amino acid is called a monomer. So that can be an amino acid, an amino acid, an amino acid. And when you link together monomers, you make a polymer. The delta S in this case is a negative delta S, and the negative plus this negative means that you're going to have a positive delta G. Is this reaction spontaneous or non-spontaneous? It is non-spontaneous, and this means that it requires energy to make this reaction occur. Okay? The reaction that is occurring is the formation of a peptide bond. The blue bond down here in this picture is showing the peptide bond between 2 amino acids. Okay? What happens is something called a condensation reaction, and in that condensation reaction, what ends up happening, so condensation means that there's water involved, you're going to lose 2 hydrogens from the second amino acid and 1 oxygen. So those 2 hydrogens and 1 oxygen leave as water, And what you're left with is the first amino acid still, it has the carbonyl group, the C double bond to an O, and now you generate a covalent bond between the carbon on the carbonyl and the nitrogen on the amino group, and that is called a peptide bond. Notice water comes out. So you can have a di, right? Dipeptide would be 2, tripeptide would be 3, etc., 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 etc. Well, basically anything less than 50 amino acids is called a peptide. Anything more than 50 amino acids is called a protein. You always write proteins and peptides with the N, right, the amino terminus on the left, and the carboxyl terminus on the right. So you're always going to go NCC, NCC, NCC. Right? That would be a tripeptide, and you would have to fill in the remainder of the atoms on that particular chain to get credit on a quiz or upload. They're showing you here, right, an amino group. So when you start linking together amino acids, you lose the carboxyl group on, right, the next amino acids, right? So here's the nitrogen. So this peptide bond means that you no longer have an amino group when you're in the middle of a peptide, but you always will still have one carboxyl group on any peptide or protein sequence. I put this in here just to give you an idea that we're talking about a string of amino acids. So you have an amino acid bound to an amino acid, bound to an amino acid, bound to an amino acid, etc. Right? You can go forever. And so this is just a string of atoms bound to one another, but they can form into any kind of structure you can imagine. And so, right, this is a little tiny structure, this is a huge structure, this has a weirdo shape. Look at this one, it looks like a donut. And what becomes really apparent the more you do biology is that the shape, right, that one looks like a dumbbell a little bit, the shape, the structure leads to function. So let's just take, for example, if I say to you, structure leads to function, what can you imagine about this protein here in the middle that I've drawn the arrow to that has a donut shape to it? What might that protein do? And look at the name of the protein. It's called porin. Guess what it does? It makes pores in membranes. So if this is the outer membrane of a cell and you have a porin protein here on the surface, it actually creates a hole. It's not drawn very well. All right, so that things can go in and out of a cell. Okay, and they specifically call these porins because they make pores. Not all proteins are as simple as that. Some are really complex, and it just depends on the amino acids and the structure of the amino acids, how they're linked together to create particular structures that lead to particular functions. And so let's begin to understand the levels of organization in a peptide or a protein. So we're talking about strings of amino acids. There are 4 levels of organization. The first level is really simple. Guess what it is? So the first, or what we call the primary structure, primary, this is another way to say primary, is just the sequence of amino acids. So here, they're showing you the sequence of amino acids of a particular protein. Okay, so this is threonine, proline, glutamic acid, okay, so that's an acid, D. TPDD, that's a sequence of amino acids in a protein. And this protein plays an important role in red blood cells. And so when the protein has this particular sequence, red blood cells look, quote unquote, normal. But if you change one amino acid in this particular protein, so now you have threonine, proline, valine, and a glutamic acid. Oh my God, okay, OMG, people. Those red blood cells do not look the same as what's on the left-hand side, the normal ones. They're actually called sickled because of the shape. They're sickled red blood cells. And the change in this amino acid in what protein, so the protein that's changed is hemoglobin. Okay, so this is just a very small portion of the sequence of hemoglobin, but we know that a single amino acid in that protein at those particular spots causes this major defect in red blood cells. And what's hemoglobin good for? It helps carry oxygen, and this is really detrimental to your health. You die pretty quickly if you have sickle cell, and it's called sickle cell anemia. Sickle, is it LE? Yeah, sickle cell anemia. Okay. So the primary structure is just, right, so the primary structure is a sequence of amino acids. Amino acid, amino acid, amino acid. It's just a string of amino acids. Secondary structure is formed, and what that means is that in these green ribbons that you see here, you get two different shapes made. One is called an alpha helix, and the other is called a beta pleated sheet, like a sheet cake. And these particular structures form due to hydrogen bonding on the backbone chain of the peptide or protein sequence. So what does that mean? Well, the alpha helix is like a spiral staircase. So if I were to, oops, sorry about that. If the alpha helix is basically like a spiral staircase, it looks much better here in the image. There are carbonyl groups, all right, that have an oxygen on them. I'm going to take that back off. Maybe I will erase that. Okay, so there's a carbonyl group that has a partially negative charge on it. That's partially negative because of an oxygen. It is electronegative. It sucks the electrons closer to it. And it will form a bond with hydrogen that's on a nitrogen that's approximately 3.6 amino acids away. All right, and so you see that you have all of these hydrogen bonds formed based on the backbone chains, right? So the NCC, the NCC, NCC. There's hydrogen bonding on the backbone structure, on the backbone atoms of the peptide chain that cause these particular structures, the alpha helix and the beta pleated sheet. In a beta pleated sheet, instead of actually wrapping around itself and creating a little helical structure, the amino acids line up side by side, flat next to one another, and once again you see these hydrogen bonds that are forming between the partially positive hydrogens and the partially negative oxygens that are bound to the backbone of the peptide chain. So secondary structure is all about hydrogen bonding of the backbone atoms. Tertiary structure. Remember I told you there are 4 levels of structure. The primary structure is just the sequence of amino acids. The secondary structure is due to hydrogen bonding on the backbone. Tertiary structure is then really directed by the different R groups on each amino acid. Remember, you have some R groups that are hydrophobic. You have some that are polar without a charge. You have some that are polar that are charged. And taken together, all of these R groups then allow for a real structure to form. So let's say, for example, in the left-hand panel of this picture below, you see I'm putting a red square around a structure that is formed by 4 alpha helices. There's a light blue, a dark blue, a yellow, and maybe a red/orange alpha helix, and they're each connected by a little loop. So the alpha helices are secondary structures, but then the 4 alpha helices fold together, and that's due to interactions between R groups. In the middle, there's a bunch of beta-pleated sheets. So think of this as you've made a piece of bread. Maybe this top piece of bread is this beta-pleated sheet, the 3 green or green-blue beta-pleated sheets, and underneath you have the green, yellow, yellow, orange beta-pleated sheets. So that's another piece of bread. So now you have 2 pieces of bread, and the interaction between these pieces of bread really are the interactions between the R groups. And even the picture on the right, it says that it's a structure that's rich in disulfide bonds. Do you remember me telling you that all of the interactions that occur that are in figure 1 on the left, or figure A and figure B, all of those interactions are non-covalent interactions. But you can also get tertiary structure due to disulfide bonds between 2 cysteine residues, right? Cysteine is an amino acid. And if you have cysteines at different places in primary structure of amino acids, then they can fold up and create these disulfide bonds, and now you have a covalently linked structure. So the issue, right, of tertiary structure is all about the things that we've talked about with respect to non-covalent bonds, van der Waals attractions, ionic bonds, hydrogen bonds, and I don't know why it's not here, but maybe I considered this, and hydrophobic force. And you have to add in the one that is covalent is the disulfide bridge. So this image shows the tertiary structure of a particular protein sequence, and what you can see, the green represents, so if I were to take green, right, and just maybe make it smaller, right, you would just have a green line of amino acids, and when it starts to fold up, you see that you've got some alpha helices I'm pointing to, and those alpha helices are due to that secondary structure, and that's due to backbone hydrogen bonding. Okay, and then you see some tertiary structure due to disulfide bonds, as well as the interaction, right? There's an ionic bond, there are van der Waals forces that really have to do with hydrophobic amino acids, so this one is valine and this one is valine. So you have 2 valines which are both hydrophobic, so they're going to want to bury themselves and try to be away from water, and they're going to be as close as they can be based on van der Waals forces. And then you can also have hydrogen bonding as part of the tertiary structure that's not part of the backbone. So this particular amino acid, let's see, the amino acid here, CH2OH, is serine, and this particular amino acid is CH2 and CC. So I think this one is, I should know better. I think this is supposed to be arginine. And the nitrogen is always going to have a partially negative charge, and the hydrogen over here will have a partially positive charge, and that creates a hydrogen bond between these R groups. So you can have tertiary structure form based on all of these interactions. Now, there's 4 levels, and the 4th level is really easy. The quaternary structure, right, it only occurs when you have more than one polypeptide chain. And what am I talking about? So any individual polypeptide chain can fold up into a tertiary structure. And then if you have a tertiary structure of one polypeptide chain and a tertiary structure of a second polypeptide chain, they can link together through non-covalent bonding. In the picture on the left showing the crow protein, it's a dimer of two of the same exact protein that are bound together here through non-covalent bonding. On the right, it's actually a cartoon diagram, right, the ribbon structure of hemoglobin, and it's a tetramer. So you have the light blue, the yellow, the green, and the purple, and there's interactions between these four polypeptide chains that create the overall structure of hemoglobin. Any one of these does not really make hemoglobin. It's a hemoglobin subunit, but you need four of those to make a true hemoglobin protein. Well, proteins play a major role in biology, and I think most of you know that you are made up of a lot of protein. We're supposed to eat a lot of protein, and it's because you have to eat proteins because we need some of the amino acids. There are essential and non-essential amino acids. We eat essential amino acids because they're essential, and we actually don't synthesize them very well. And so we use the amino acids that we eat to create proteins that have these different functions. Some proteins provide defense for our body, antibodies, so AB stands for antibodies, and complement is another part of our immune system. Those are all proteins that play a role in defending us against outside intruders. There are proteins that play roles, which we learn about in Biology 380 in gory detail, that allow for the movement of materials inside of cells. There are proteins that play roles that signal from one cell to another. Hormones are one of those. And if you don't know what a hormone is something that signals. So think of testosterone, estrogen, what else? So there's a lot of other hormones that play roles. I'm trying to think of thyroxine. These are all hormones that are secreted by one cell and they interact on another cell. Insulin. Proteins can also provide structure for cells, either mechanical support as in the cytoskeleton. There's proteins that are part of cytoskeleton, but also structure in creating the shape of even the nucleus. The shape of the nucleus is determined by cytoskeleton. And then there's transport molecules, and probably the biggest, most important class of proteins are those that are enzymes. And what enzymes do is speed up reactions, and we're going to talk more about enzymes, but think of enzymes as they bind to the substrate, and that helps to change the conformation of the substrates that makes the reaction proceed in a faster manner. And that we call catalysis. So enzymes catalyze reactions. This whole concept of catalysis is very important because the enzymes, as I said, will bind to substrates, changing their conformation, and what that does is it allows the substrates to interact in a completely different way than they would otherwise, and it lowers the free energy state of whatever the intermediate in the reaction is. Let's look at delta G with respect to a... And so here in this exergonic reaction, this is a reaction with a negative delta G, which means it's spontaneous, and it does not require what? Energy. Doesn't require the input of energy. And you have reactants or substrates that have a particular free energy. Here we have A, the molecule A, that's going to be interacting with a molecule of BC. And if you skip over the middle of this graph, what you see is the reactants end up going to the products. And if you look at the products, look what happens. Okay, now you have A is connected to B, and C is now separate. And the delta G of this reaction, all right, goes from the level where I've drawn the red line to the level, right, so the arrow is going down, so that makes it a negative delta G. And the only thing that determines delta G is the difference in free energy of the reactants compared to the products. But what's that big bubble in the middle? Well, it actually is hard to make this reaction occur. You have to go uphill. You have to increase the free energy of what we call the intermediate. So the transition state is the intermediate in the reaction. And we do that with something that's called activation energy. All right, here's my typical example of this. If you're lying on the couch, right, it takes some energy for you to get up off that couch. Would you agree? But you are not at your lowest free energy state when you're lying on the couch. You could actually be at a lower free energy state by lying on the floor. But to get from the couch to the floor, you actually have to put a little bit of energy in to push yourself off of the couch to reach that lower energy state. So the activation energy would be you putting a little bit of energy in to, right, push yourself off the couch, and that then leads to the delta G being negative because you actually went to a lower energy state. Well, notice what happens here. The first line that I've just drawn an arrow to has a large activation energy, and it might be hard to overcome that, but when you add an enzyme into the reaction, the activation energy, the EA, the energy of activation, is decreased, and it's because you have the binding of that enzyme to the reactants, and it changes their shape, and that allows for the reaction to proceed. Notice the delta G is exactly the same because you have the same reactants and the same products. They end up at the same place. The difference in free energy is the same as with or without the enzyme. Kind of an important part of understanding enzymes was the finding by Fisher that he called it a lock and key model because he said you had the lock, all right, was the enzyme. You put the key in, and the key were the substrates that go into, right, a key only will fit into the correct lock, and then when that lock binds to the substrates, it changes the shape of the substrate just enough to change what happens, and that opens the door. And so notice what happens here is we're saying that this in-between state, when you have the enzyme bound to the substrate, you have the intermediate, you have the transition state, and then once the reaction occurs, it changes, right, the conformation, and now you have totally different products that come out. What happens to the enzyme in this? Absolutely nothing. It ends up exactly the same way as it started. So enzymes are not consumed in these types of reactions. Enzymes can be reutilized over and over and over and over and over again, as long as, right, a protein is still good. Think about it. What is protein? If you take a piece of meat out, you can eat that piece of meat if you cook it. Right? You could eat it without cooking it, but if you eat that piece of meat, you know, if you cook it in 5 minutes, it's going to be fine. If you cook it in 20 minutes, it's still going to be fine. If you cook it in 24 hours, that's not so good. Proteins don't last forever. So, right, an enzyme will last over lots and lots of rounds of catalysis without any problem. Here, this is showing you how the transition state might alter the enzyme itself. And so they're showing you on the top a substrate that is going to bind into, right, the lock, and as it does, it's almost as if the enzyme itself grabs onto the substrate. There's a change in the structure, and the change in the structure actually allow for changes in the binding of the substrates, and that leads to the lowering of the activation energy. Don't worry too much about this bottom part. It's actually showing the atoms and how they interact with one another. But how would you suspect temperature? All right, if you increase the temperature, what's that going to do to an enzyme reaction? What happens at higher temperature to atoms? They all move faster, and so you're actually going to maybe increase the speed of a reaction. What about pH? That might change the speed or the conformation of a protein, so that might also have changes in the function of an enzyme. And so enzymes play a really important role. Now, that first example was showing you an exergonic reaction where you had a negative delta G. It was spontaneous, but what happens when you have a positive delta G? That means that your substrates are going to start out at a free energy that is below the free energy of the products. And so the delta G in this particular example is a positive delta G. That means it's non-spontaneous, and when it's non-spontaneous, you have to add energy into the reaction. In this one, they're showing you that by having ATP around, you're able to break the bond of ATP. So you break off one phosphate. Now you have ADP in this inorganic phosphate. But the energy from breaking that bond is added to this reaction, and that's where you end up with the positive delta G. So the only way that reactions that have a positive delta G can occur is by putting in energy that helps the reaction go forward. Where does this fit into our entire idea of, can a protein be its own, right? Can it be the first self-replicating entity that happened in the world? It actually can't. It has some of the features that would be necessary. Okay, the first self-replicating entity had to be able to replicate. That requires catalytic activity, and we know that proteins can be catalytic, but proteins do not contain enough information to be the hereditary material. So, right, so we'll put a big circle through that. So proteins were not the original self-replicating molecule that was the first life form. And I think that a bit about covers it for proteins. We'll pick up our next lecture talking about nucleotides and continue on from here. Have a good weekend.